Okay, we are back with the rest of chapter 16. Remember earlier we were just talking about how DNA is going to be wrapped around. DNA is mostly negatively charged. It's going to be wrapped around these histone molecules in order to be wrapped up in nice little packages called chromosomes. Okay, so within our DNA, specific structures code for specific proteins and for specific traits, we'll say. And it's the reading of those and the activation of those specific traits, those traits that we call genes, that determine everything about us. Determine the functioning of our of all of our proteins inside everything, as well as down to things like hair color, eye color, all that sort of stuff is all coded by genes, and depends on like how the proteins and certain things are produced. So covalent. So we're talking about the regulation now of the genes, because some genes we want to be turned on at certain points or turned off at certain points. Some we want to be expressed at very specific levels, and so we have to have a way to properly regulate and control all of the genes. So we're going to talk about covalent modification of histones by acetylation and deacetylation. So those histones that are wrapped around the DNA, holding all the DNA in place, those are going to be modified by acetylation and deacetylation in order for the genes to either be expressed or essentially turned on or turned off. So reading here, so histones play an important role in gene regulation. Gene expression is influenced by covalent modification of histone proteins. Histones can be modified covalently by methylation, acetylation, glycosylation, phosphorylation, ADP ribosylation, and others. So in an inactive gene, or when the gene is turned off, the DNA wraps around the histones, and the nucleotide sequences are hidden and cannot be read. So think of it like the histone just like grabbing and holding and hugging onto the DNA, making it so that other proteins can't come in and they can't read that strip of DNA. They can't read that specific code, which codes for specific proteins that will go off and do a specific function. So during gene expression, that's talking about an inactive gene, right? The histone is hugging it really tight. During gene expression, the DNA molecule must be unwound to reveal the hidden or inaccessible portion of the nucleotide sequence to be transcribed. This occurs when the histones are detached or shifted, that causes reading of the nucleotide frame related to that gene expression. So we're going to bind something to the histone. Remember when we talked about things binding to proteins, you can have changes in the shape of the proteins. And that's what's happening here. When the histone gets something bound to it, its shape is going to change, and it's going to essentially release the DNA, or it's going to slide up to a different part of the DNA to reveal the gene code that is then going to be read. RNA can be produced. That RNA can then go be off, and so it's going to transcribe RNA sequence, and that RNA sequence can then go off and be translated and have a protein be formed, or have an amino acid chain which turns into a protein be formed from that. So when the histones are covalently modified by acetylation, catalyzed by histone acetylase, good little enzyme to remember there, It relaxes or unwinds the DNA. The relaxed form is also known as euchromatin. And you should be familiar, this is a term that you should have learned in BCT as well. So around that specific gene, which is now exposed and can be read by the RNA polymerase, leading to transcription. Acetyl-CoA is the donor of the acetyl group. Acetylation occurs at the lysine residues of histones. Acetylation eliminates the positive charges on histones and thereby decreases its interaction with the DNA, and it unwinds. Deacetylation of histones by histone deacetylase enzyme brings the DNA back to its original histone-bound form, so it condenses it to form heterochromatin. Euchromatin, relaxed, open, heterochromatin, tight, condensed, which then again causes suppression of the gene expression. Numerous studies have shown that the presence of H1 inhibits transcription, and in some cases, transcriptional activation is associated with the removal of H1 by its phosphorylation, thereby decreasing the affinity of H1 to the DNA. However, some studies also indicate presence of histone H1 in transcriptionally active genes, even though in an altered chromatin organization. The link between histone H1 and core histone acetylation is not clear, so a question mark is shown in the diagram for this. Don't worry too much about that last part. Focus on what I have highlighted specifically up here. So we have the histone octamer. It's all wrapped around the DNA. The histone acetylase binds that acetyl group coming from acetyl-CoA, and it relaxes and it opens it up and then allows for the RNA polymerase to bind and do its thing. So DNA methylation. Within the DNA sequence, there are many cytosine guanine repeats or CpG islands. The presence of CpG islands in the 5' end of some genes are related to methylation and regulation of gene expression. So there can be a repeating sequence, cytosine guanine, so it has a repeated sequence there, kind of like a marker to indicate, hey, methylation is going to happen here. The CpG-rich islands in nucleosomes have decreased levels of histone protein H1, probably indicating DNA packaging is relaxed. Other histones are acetylated in this region, indicating a favorable condition of gene expression. In numerous cases, this CpG-rich islands are localized at the beginning of the upstream promoter region of the gene. The upstream promoter region, so think about it upstream, so ahead of the gene, so before the gene actually, like the code for the gene is actually there, we have what's called a promoter. That promoter is going to be what, when it's exposed, what it's going to kind of help signal and identify, hey, like this is where the gene starts. This is the specific code that we need to make sure that we copy in order to express this specific gene. So DNA methylation is a process by which primarily the cytosine residues in the CpG-rich islands of the DNA sequence are methylated. It is epigenetic modification of DNA molecule. This cytosine residue methylation produces 5-methylcytosine catalyzed by DNA methyltransferase enzyme and causes silencing of a gene. So you could get a question about that. DNA methyltransferase enzyme causes silencing of a gene as a result. So as a result, the gene is not transcribed. In normal human cells, about 70% of CpG repeats are methylated. So when they're methylated, that means that whole area is closed down and we're not going to get that gene expressed. Methylation of DNA protects genes from nuclease activity, which breaks down DNAs, and thus also increases DNA stability. Demethylation of the 5-methyl-cis cytosine residues causes hypomethylation. Hypomethylation, or removal of methyl groups from the cytosine residues in the CpG-rich areas in the promoter region of the gene by demethylation, can result in increased gene transcription. Decreased levels of DNA methylation can stimulate abnormal gene expression. Increased mutation of certain genes resulting in uncontrolled gene expression are often seen in cancer tissues. So we want our DNA to be properly methylated. If it's hypomethylated, bad things can happen. We can get abnormal gene expression, and as a result, you can also see it with certain with a lot of cancer tissues. Okay, now we're talking about denaturing DNA and renaturing DNA. So denaturation of DNA, so changes in pH, ionic alterations, and temperatures can interrupt the base pairing properties in DNA. So the extremes of the pH, like strong acidic or alkaline solutions, and also heat, cause DNA strands to be separated without breaking the phosphodiester bonds. So that's going to affect or disrupt the hydrogen bonds, which is the basis of base pairing between the two strands. Renaturation of DNA, also called annealing. Heat denatured separate strands of DNA, undergoes a slow decrease in temperature, and that causes the reformation of the base pairs, complementary strands of DNA, all coming back together. So denaturing DNA, breaking it apart, annealing or renaturation, it coming and forming back together again. As long as it's partially, you can eventually have, like, if you put it in like extreme enough temperatures or extreme enough pH or specific solutions, you can completely and irreversibly denature DNA. And we talked about that briefly in cells and tissues as well. So hopefully this is a little bit of a review in this chunk here. Hybridization. So a single strand of DNA or RNA pairs with a complementary base sequence on another strand of DNA or RNA. So when you denature a bunch of different strands, when they're denatured and kind of free-floating, you mix them all together. As they cool, they can end up combining and recombining in different orders and different matching with different original strands, giving with complementary base sequences on the other side, which just gives you essentially different types of strands. And it's called hybridization. Or actually, strands with DNA or RNA from two separate sources that are then combined. So DNA biosynthesis or DNA replication. So getting into replication, pay really close attention to a lot of these definitions, as these are going to come up quite a bit in questions. So replication is semi-conservative. What this means is that each DNA strand serves as a template for the synthesis of a new and complementary strand. So thus each original strand synthesizes two new daughter strands with complementary base pairs. So each original or parent strand synthesizes two new daughter strands with complementary base pairs. So following replication, each daughter DNA contains one intact parental strand and one newly synthesized strand joined by base pairs to the parental DNA. DNA replication is also bidirectional. So replication starts at a site of origin and simultaneously moves out in both directions from this point. So semi-conservative, this is talking about being semi-conservative here, so it means it semi-conserves that parent strand. So these two strands are originals. They're going to separate and get copied, where we have a, not copied, but have a complementary strand built for each of them. And now we have 2 sequences of that DNA. Now these are also going to get broken up later on down the road and copied again. So see how we're conserving some of the original DNA, but also we have these 2 strands are completely new synthesized strands of DNA, while we still have the 2 very original strands from the original parent molecule. Talking about bidirectional DNA replication. So replication begins at an origin, and a new strand of DNA is synthesized from 5' to 3' direction. Remember that. This is key. That DNA is synthesized from 5' to 3'. Always 5' to 3'. So during addition of the nucleotides, the second nucleotide is added to the 3' OH group of the first nucleotide. Thus, the synthesis moves from 5' to 3' direction, and the chain is elongated at the portion of the DNA is open or relaxed. In the leading strand, the synthesis is continuous. In the lagging strand, the synthesis is discontinuous. So let's break this up a little bit. So we have our strand of DNA, just like that. Okay, and all this is bound. There's all these hydrogen bonds binding everything there. The hydrogen bonds are going to be broken at some, let's say it's broken here, where it opens and starts. So it opens here, and we start to copy the DNA. So it starts to unzip towards this direction. And that's what we're specifically looking at right here. So we have the leading strand. So remember, we're always going to replicate DNA in the 5' to 3' direction. So our leading strand can just attach and go straight on through. Whereas our lagging strand is going to make something that's called Okazaki fragments, where it's going to make these little chunks, 5' to 3', 5' to 3' until it hits the next one, and then the protein synthesizing will pop off, and later on something will come along and splice these ends all together to make them work. So you have the leading strand that goes straight through, 5' to 3', and lagging strand that is discontinuous. And we'll get a little bit more into that down here. So DNA replication. So let me actually see if I can find this. Doesn't go into a lot of I know that in NBCE we talk a little bit more about this. If you don't quite remember a lot of this process, I would recommend looking up a quick YouTube video about the synthesis, or like the replication of DNA. Let me see if I can pull. I remember these ones are usually pretty straightforward. So when you have DNA wants to divide, let's go through all of this. It's going to show about when it's going through. So we have our different enzymes: DNA polymerase, helicase, primase, and ligase. They're all going to have different steps. So helicase is the unzipping enzyme. It's going to go through and unzip the 2 different strands and help with unwinding of it, breaking those hydrogen bonds. DNA polymerase comes in. It's going to bind, and it's going to put down an RNA primer, because you need some place for the rest of the molecules and everything to bind. Yeah. So I would take a minute, go through a video or something like this, just to look at these, refresh your memory with these different molecules. So that way you can just remind yourself this process as a whole. It'll help you remember just what is specifically talked about here. Okay. So, DNA replication. In the leading strand, DNA synthesis proceeds by separation of the parent strands, followed by unwinding of the DNA double helix ahead of the replication fork in a 5' to 3' direction with a free 3' OH group, and is continuous. In the lagging strand, the DNA synthesis is discontinuous. DNA polymerase 1 catalyzes DNA synthesis, so that unwinding and everything before, that was the helicase. DNA polymerase 1 catalyzes DNA synthesis. DNA polymerase 1 enzyme copies or reads the parent strand template in the 3' to 5' direction, and the synthesis occurs from 5' to 3'. As the synthesis proceeds, phosphodiester bonds are formed between the nucleotides. DNA polymerase 1 requires a single unpaired template strand. All 4 deoxynucleotides, so all these have to be present in the area so it can build. An RNA primer strand of 10 nucleotides length with a free OH group at the 3' end, to which a new nucleotide unit is added. Synthesis of this RNA primer occurs using the parental DNA template. The deoxynucleotides are added to the 3' OH group of this RNA primer. The reaction product has a new free 3' OH group, allowing addition of another nucleotide. So here we have mechanism of elongation, showing you the building as it goes, as it's connecting, building all the way through. Again, reading in the 3' to 5' direction, building 5' to 3'. DNA synthesis in the lagging strand. In the lagging strand, DNA synthesis occurs via formation of a series of Okazaki fragments. As the replication fork opens, in the Okazaki fragments, RNA primers are attached. Later, as multiple Okazaki fragments are generated, replication complex begins to remove the RNA primer and fill in the gaps left behind by the removal of the RNA primer. The gap is filled in by attaching the right nucleotide followed by joining, or sealing the gaps by DNA ligase. So we're going to have these little primers built so that DNA polymerase can do its job and copy these chunks, and then it's going to pop off, and then DNA ligase comes through and finishes off the strands, takes off the primers, and finishes filling in the last of the DNA. DNA ligase later joins Okazaki fragments and completes the complementary chain. DNA synthesis uses an RNA primer synthesized by primase or RNA polymerase at the site of initiation of DNA replication. The RNA primer is subsequently removed by the 5' to 3' exonuclease activity of a DNA polymerase. The DNA polymerase fills the gap by choosing and adding the right nucleotides. DNA ligase joins the gap in an ATP-dependent manner. In the mammalian nuclear genome, all the RNA primers are removed at the end of the replication process. However, in the mitochondrial genome, small pieces of RNA primer remains. Here we go. We're getting into the order, and this is a little bit weird, but it all kind of fits. So the DNA helicase unwinds the DNA helix ahead of the replication fork. The topoisomerase acts to prevent the extreme supercoiling of the parental helix. Topoisomerase breaks and rejoins DNA chains, and topoisomerase is also able to unwind supercoiled DNA. So kind of doing an overall idea of this. apologize if this part isn't perfect, but I'm going to go as much of my memory as I can. So we have our DNA, that's here. We're going to have our DNA helicase come through and break all of the hydrogen bonds. While it's doing that, like if you ever have like an extension cord or something that's coiled, as you start to uncoil part of it, like a hose is coiled, you notice that as you uncoil one end, the rest of the cord or the hose gets really, really coiled. So topoisomerase, what it literally does is it'll come and it'll like break a bond here to let it uncoil, relax a little bit, and then reforms it. And then it breaks another bond, helps to uncoil, helps it relax, and then rebond. So it prevents that, so what's called supercoiling. So the helicase comes down breaking those hydrogen bonds. Topoisomerase is doing that job. Then we get to a point where it opens it up to allow for copying. So we have an RNA primase. Where is it? So by primase, so there's two possible enzymes, primase or RNA polymerase. We have, once this is broken up, just continuing down here, so we have two different strands here. These are both open. This is our 5' to 3' end. This is 3' to 5'. Let me just flip those. No, it'll be fine. It'll be fine actually. So in this direction, 3' to 5', this is going to be our leading end, and this is our lagging end. Because we get DNA formed 5' to 3'. So our primase or our RNA polymerase is going to come and set up a primer. This is going to allow DNA polymerase to come and bind and head down its merry way, building a second strand of DNA, 5' to 3'. And that's just going to keep on going until we've replicated this whole chunk of the strand. The lagging end, because it has to go 5' to 3', so we can't just do the same thing down this way. So we're going to have a primase, build a little primer, build a primer, build primers. So that way our DNA polymerase can bind and build in the 5' to 3' direction. And build those new chunks of DNA. Just like that. So this one down here, this one is all good. Good to go, easy peasy, new strand of DNA. The lagging strand, however, has all these primers and everything in here. Part of the job of the DNA polymerase is actually going to be ripping up and getting rid of this primer. So it gets rid of that primer, and essentially so we're left with something that looks like this. This is a 5' to 3'. So we're left with pretty much complete, but not quite totally complete fragments of DNA. Our DNA ligase is going to come in and finish fusing these gaps. And then we're left with two perfectly usable, good copied strands of DNA. Hopefully all of that made sense. Broke it down a little bit, made it a little bit simpler. Okay, so with DNA, this whole process doesn't always work as smoothly as we would like it to, so sometimes there are mutations. So a DNA mutation is a permanent change in the DNA nucleotide sequence, and that is known as a mutation. So these alterations are permanent changes in the base sequences of the daughter DNA. Mutations are due to either uncorrected errors made during replication or due to damage due to radiation, toxins, or chemicals, etc. That's why when you hear something is cancer causing, that's what it's referring to. It's referring to the fact that it's going to get in and mutate your DNA, which can then, depending on where the DNA is mutated, if it gets mutated beyond a point that you can, that your body can naturally fix it, then that's when you get cancer and certain diseases. So if the mutation is occurring in a non-essential portion of the DNA sequence, or if it has negligible effects, it is known as a silent mutation. So different types of mutations. We have a point mutation, which is substitution of one base for another. An insertion mutation, which is the addition of one or more nucleotides within a DNA sequence. You have a deletion mutation, which is the removal of one or more nucleotides from a DNA sequence. And then in order to repair those, our body has specific repair steps. So we'll have removal of the segment of DNA that contains a damaged portion or mismatched bases using an exonuclease or endonuclease activity. Filling up the gap with a DNA polymerase, and then ligation of the newly synthesized segment to the remainder of the chain. The enzymes that are required for DNA repair, we have an endonuclease, so DNA glycosylase enzyme, which is involved in base excision repair, recognizes DNA lesions, removes the affected base, leaves behind an apurinic or apuridinic site known as AP site. AP endonuclease cut at this site, and a segment of DNA sequence around this is removed. So it's kind of like the DNA glycosylase enzyme is marking. It recognizes where there's a lesion, where there's something that's not supposed to be, and it marks it. It says, hey, this isn't right, come back and check this. So that way DNA polymerase 1 can come back through, an AP endonuclease comes along, recognizes that cue, snips it out, and then DNA polymerase 1 slides down and will add in the proper sequence. Exonuclease, so this enzyme functions by cleaving deoxynucleotides one at a time from the end of the DNA nucleotide sequence. Breaking of phosphodiester bonds at either the 3' or the 5' end occurs by hydrolysis. So it can have 5' to 3' exonuclease activity, as well as 3' to 5' exonuclease activity. Think of this one more rather than like snipping it in the middle. It's kind of like just hitting backspace. Like you mess up at the beginning of a sentence, and you just hold backspace until you get to the issue, backspace it up, over the issue, remove the issue, or the misspelling or whatever, and then retype what you had already done. DNA polymerase, this enzyme is involved in synthesizing deoxynucleotide polymers, or simply responsible for DNA synthesis by adding deoxynucleotides and elongating DNA chains. And the DNA ligase, we talked about this one already. This enzyme catalyzes closing of the nicks or gaps in the phosphodiester backbone of the DNA. This enzyme is also needed for the joining of Okazaki fragments during replication. Okay. Sweet. So hopefully a lot of this was review. If you're still confused at any part of this, please go back through this. Go ahead and look up another video on DNA replication. Ninja Nerd, I know, always has great stuff. I know he has great stuff on DNA replication if you need some refreshers. Take the time to go back through, understand this. Know what 5' to 3' or 3' to 5', know what that all means. And like which direction things are happening, because that will be a decent chunk of this upcoming test. being able to recognize and understand what does 5' versus 3' and all that sort of stuff mean. Okay. Good luck with the rest of. Good luck with this chapter, and good luck studying, and have a great day.